Okay, so this week we're gonna cover muscles and how they work. To be honest, like this is probably one of the more conceptually dense weeks. Um, like the topic that we're gonna talk about or try to cover today is basically how neurons work and how neurons control muscle function. So just to like put this in perspective, like we just kind of covered the same thing at my school, at my medical school, and it took us like 5 hours to cover all the nuances. And so I tried my best to like distill it down so that we can cover it within the hour. But don't be like frustrated or scared if you don't get it the first time around, just because again it's a very conceptually dense topic. And if you looked at Moodle, there are a lot of videos to kind of help you better understand like what is happening. But again, like I would say use the videos, definitely do those knowledge checks and exercises, and you'll see that the depth of questions that we're asking aren't really too hard or too stressful. It can definitely be hard, but we're trying our best, Ollie and I, to make this more palatable. So let's just jump into it and kind of see what happens, I guess. So this is our introduction to the nervous system, and it's a three-part chapter. So we're going to cover the first part of the nervous system because you really have to understand how neurons work in order to understand how we have voluntary muscle contraction. And so you can see here the hierarchy of the nervous system. The nervous system is broken down into two separate systems. One is the central nervous system, which is going to be the brain and spine, which we'll talk about probably next week. And then you have the peripheral nervous system. And this peripheral nervous system is basically everything but the brain and spine. It's all the spinal nerves that emerge from the spine and the brain. And these spinal nerves are either autonomic or somatic. So autonomic means it kind of sounds like automatic, right? Autonomic, automatic kind of sounds the same. So this means that these neurons are controlling organs and glands, right? Because you don't really think about contracting your heart, right? You don't think about secreting insulin into your blood, right? All that stuff is handled for us automatically. And the autonomic nervous system itself is broken down into sympathetic and parasympathetic systems. So sympathetic, you guys might have heard me mention it, is the fight or flight system. So this system is kicked into overdrive whenever you're trying to escape a threat. And then for the most part, we spend a lot of our time, a majority of our time, hopefully, in this system, the parasympathetic system. This is called also the rest and digest. So right now, hopefully, we're all in this kind of phase, right? And this stuff we'll talk more about probably next week or so. We're going to talk mostly today about the somatic nervous system, namely the motor part of it. So basically how we contract our muscles, right? So somatic, or whenever you see somatic, it means body. So basically this is the part of the nervous system that allows us voluntary control over our skeletal muscles. So let's talk a little bit about what a neuron is. A neuron is a specialized type of cell, and the purpose of a neuron is to transfer information from one direction to another, from one location to another. So for example, when you think about like contracting any muscle, right? When you think about clenching your fingers, making a fist, right? That signal is traveling from your brain all the way down to your spine, and then from your spine to your actual fingers are the muscles of the forearm, right? And the muscles of the forearm are the ones that are contracting and tugging on the tendons that are in your fingers so that we can actually move our fingers around, right? So it's basically like these cells either send information to each other, or they send information to muscles, or they can also send information in glands. So that's the purpose of these neurons. And you can see that the way they're designed kind of tells you that that's what they're meant to do, right? Because in anatomy and physiology, for the most part, structure and function are related to each other. So for example, if we look at this neuron, right, you have the head of the neuron that we call the cell body. And the cell body is also where you're going to find the nucleus. And then you can see all these crazy branches coming out of the cell body. We call these the dendrites. And so this part of the neuron is the input, right? And the input is how this neuron gets information from other neurons. The thing about neurons is that they always work in collaboration with each other. So even though only one is shown here, I want you to realize that there's always a network of neurons, and they're all trying to influence each other's actions, right? And the input for any neuron is here at the dendrite. Right? See the dendrite here. And so once it collects this information, right, in the form of neurotransmitters, which we'll talk about, once it's collected this information, it will then accumulate it and make the decision to either fire or not fire an action potential. So an action potential is this flow of current that travels down the axon of the neuron. And we'll talk about that as well. So let's switch to slideshow. Okay, so the action potential, or also known as the flow of currents, of positive currents, will travel down the neuron and then gets to the terminal, right? So this is the output part, right? So the output part allows this neuron to communicate with either a muscle, a gland, or other neurons. And the reason why this is the output part is because this is the part that releases neurotransmitters. Right? You guys probably have heard of neurotransmitters. You guys heard of like serotonin, dopamine, norepinephrine, like those are all like neurotransmitters, right? Noradrenaline, GABA, glutamates. Those are all potential neurotransmitters that a neuron can release. And those neurotransmitters will then bind to the dendrites of another neuron, for example. So it basically is designed to communicate with either neurons, glands, or muscles, right? So to reiterate, a neuron may terminate on any of these three structures, right? It can terminate on another neuron. And when it terminates on another neuron, right, when it releases neurotransmitters on another neuron, we call that a synapse. We're going to talk about this today as well, right? When this neuron terminates on muscle, we call that neuromuscular junction. The space where they interact is the neuromuscular junction. When neurons interact with each other, the space that they interact is called the synapse. We're not going to worry too much about this. We'll probably talk about it later on when we talk about the endocrine system. So it's mostly just these two for now. So when a neuron transmits or terminates in a muscle or gland, so terminates just means that it just releases neurotransmitters, all right? So when a neuron releases neurotransmitters on a muscle or a gland or even another neuron, the neuron says to innervate that structure. So innervate is the same thing as stimulate, right? So stimulates, innervates, it's all the same thing. So we're just getting more familiar with some of the terminology that is presented to us. And this guy here, called a motor neuron, these are the neurons that will innervate or stimulate muscle fibers, right? So motor neurons are neurons that will stimulate muscle fibers. So keep in mind that there are a bunch of neurons, right? There's some neurons that will stimulate a gland and tell that gland to release a hormone. There's a bunch of neurons that will innervate and stimulate a muscle, telling that muscle to contract. And the neurons that do that are called motor neurons, right? Think of like motor movements, right? We're trying to contract muscle, we're trying to move our skeleton. And so the first thing that you should know about a neuron is that there's a higher concentration of sodium on the outside of the neuron relative to the inside, and a higher concentration of potassium ions in the inside of the neuron relative to the outside. So this is something that you should just have like memorized. It's a core concept in physiology. You have this neuron and surrounding it is this extracellular fluid or extracellular space. So extra means outside, right? The environment that it's in will have a higher concentration of sodium and a lower concentration of potassium. Right? So the environment that this neuron is in is always going to have a higher concentration of sodium, lower concentration of potassium. And this has repercussions. Right? So the first question is, if sodium is able to enter the neuron with the aid of the concentration gradient and the sodium protein channel, like what kind of transport is this? Right? So this goes back to like that week when we talked about like cell transport. That information will always show up like throughout our organ system. So it's essential that you still remember what diffusion means, what facilitated diffusion means, what primary active transport means. If you don't remember that, it's going to make it very, very hard to continue onwards with physiology in general, right? So we're going to try to like keep bringing it up so that it stays kind of fresh in our memory. But why don't we attempt this first question and kind of see what we remember. I'm gonna open the poll up. All right, so I'm gonna close that and we'll talk about this together. So as a brief review, right, let's go over all these definitions together. So this question mentioned that sodium is able to enter the neuron with the aid of the concentration gradient and the sodium protein channel, right? So there's two key pieces of information that will let us know what kind of transport this is. So first of all, the sodium is going from a high concentration to a low concentration, right? So a high concentration to a low concentration. So it's going with the concentration gradient, right? And remember, this was a passive process. This is a passive process. So that way we can kind of get rid of C. We know C isn't true because primary transport is active, right? So what is primary transport again? Primary transport is when we move things from a low concentration to a high concentration, right? So that would be primary transport. But this isn't the case here, right? We're observing that sodium is moving from high concentration outside and it's trying to move to a lower concentration of sodium inside, right? So that lets us know that it's some type of diffusion, right? So that's what diffusion is. So the question now is it regular diffusion or is it facilitated diffusion? So the word facilitated means to help, right? So remember that facilitated diffusion involves some type of protein channel, right? So facilitated diffusion involves some type of protein channel. And this question explicitly states that sodium can only move in to the cell with the aid of a channel, right? So this will be a facilitated diffusion, right? It's not regular diffusion. It's not regular diffusion just because there's a protein channel involved, right? Regular diffusion just states that it goes from high to low concentration, but it does so without any help, right? But here, this sodium ion needed help, right? It needed some kind of protein channel to enter the cell, right? Remember that some things have an easier time entering the cell and some things need channels to enter the cell. Things that are charged, like sodium and potassium, they need channels to enter the cell, right? So just a re-summarization of previous chapters. And so the same can be said for potassium, right? So potassium is going from a high to low concentration. So what kind of transport would this be? Again, facilitated diffusion. Because as we'll go through and learn, the only way for these ions to escape or to enter the neuron is through a protein channel. So let's talk more about a synapse, right? So a synapse is an interaction between neurons. So here you have a neuron, you have another neuron, you have three neurons, right? You have 1, 2, 3 neurons. So this neuron here, we can see that it's cell body, whereas these two neurons off to the side, you only see the axon and the axon terminal. So the axon terminal is what we call the output part of the neuron, right? So here, if we zoom in, here's what we're gonna see, right? So these are the neurons, and again, you see this space here, right? So we zoom in even more, you notice that there is this space here, right, between these two neurons. And this is called the synapse, right? If we look off to the side of this diagram, the space between two neurons is the synapse. Sometimes it's also called the synaptic cleft. And then before the synapse, you have the presynaptic neuron, right? Pre as in before, right? And then after the synapse, you have the postsynaptic neuron, right? And so it should be obvious that the postsynaptic neuron is receiving input from the presynaptic neuron, right? So these knobs are supposed to represent the cell body, right? This is the cell body. This is the cell body of the postsynaptic neuron. This is the cell body of the presynaptic neuron. And again, this presynaptic neuron is releasing neurotransmitters onto the postsynaptic neuron, right? So that's what we see here. If we zoom in enough, we can kind of see the synaptic cleft. We see the presynaptic neuron, we see the synapse, and then we see the postsynaptic neuron. And what you're seeing is that this presynaptic neuron is releasing neurotransmitter, and these neurotransmitters will bind to the receptors on the postsynaptic neuron, right? And this is going to elicit some kind of change. And we're going to talk about this change, but it's important to understand what a synapse is from this figure, right? So make sure you know that the synapse is like this space, this point of interaction between two neurons. The neuron that comes before the synapse is presynaptic. The neuron that comes after the synapse is postsynaptic. And so here's a summarization of what I just said, right? Synapses are junctions between presynaptic, postsynaptic neurons. So you can see here, here's another example that can actually get kind of crazy because this axon, you can see, kind of branches out, right? These are all terminals from the same neuron, right? So the axon kind of branches out and is able to innervate multiple points on the postsynaptic cell, right? So this is the postsynaptic, presynaptic, and you see multiple synapses being formed, right, between the presynaptic and postsynaptic cell. And there's two types of synapses. There's excitatory synapses and there's inhibitory synapses. And what we're going to learn is that excitatory synapses will lead to the generation of an action potential, whereas a inhibitory synapse will shut down that neuron, right, or silence that neuron. And so there's two types of synapses, and they're dependent on the type of neurotransmitter that is released. Right? So that's as much information as we're going to go into. Just know that some neurotransmitters will excite a neuron, whereas some neurotransmitters will inhibit a neuron. Right? Some will excite and some will inhibit. So for example, alcohol, you don't have to know this, but alcohol, drinking alcohol causes the release of inhibitory neurotransmitters such as GABA, and basically those neurotransmitters will shut down neurons, right? So that's why when like you drink, you notice like you have lower inhibition, right? You don't think as much, right? That's why a lot of people kind of use it to socialize. A lot of people with anxiety, they use alcohol as a coping mechanism because it shuts down those neurons, right? It causes them to be inhibited. So GABA, which is released through drinking alcohol, is a type of inhibitory neurotransmitter. And it will be found at these inhibitory synapses. So two general types of synaptic transmission. There's electrical and there's chemical. So electrical meaning that action potential, right? Meaning action potentials. We're not going to talk about the ones that involve gap junctions, so you can cross that out. But electrical signal will travel down the presynaptic neuron, and that action potential has a purpose, as we'll soon discover. The action potential will trigger the release of neurotransmitter. Right? The purpose of an action potential is to trigger the release of a neurotransmitter. Right? And again, we're going to go into more detail about this, but I want to kind of warm you guys up to this idea. So action potential travels down the neuron. That action potential will trigger the release of neurotransmitters. Neurotransmitters will then act on this postsynaptic neuron. Right? So the chemical part of it comes from the neurotransmitter. So that's why we often say that the interaction between neurons is both electrical and chemical. Electrical meaning that there's like current involved, right? The signal travels within the neuron. And then once it gets to the end of that neuron, the electrical signal will trigger the release of the neurotransmitter, which is the chemical. And the neurotransmitter will bind to neurotransmitter receptors on the post-synaptic cell and cause that cell to do something. Right, so here, again, is just terminology. This is like a lot of terminology for the most part, most of which we already mentioned. Yeah, so exactly right. So drinking, that's why drinking does make some people sleepy, right? And like as you get older, it makes you sleepier. And that's totally anecdotal. So again, just to cover some of this terminology, hopefully some of it is starting to sound more familiar. There is a delay. It takes time for the electrical signal that's traveling within the neuron to make it through that neuron and to release neurotransmitter. So there is a delay, but the delay is very trivial. It's like 0.5 milliseconds, right? For the electrical signal to travel down an axon and to trigger the release of these neurotransmitters here. So that's called synaptic delay. So presynaptic neuron, a neuron that will be conducting the action potential towards the synapse. So if we look at this picture, again, you have your synapse, right? Your synapse is here. I'm just writing an S for synapse. And synapse is a space between the presynaptic neuron, right, and the postsynaptic neuron. That's what you're seeing here. The presynaptic neuron is a neuron that fires the action potential, right? You see the red line indicates, or this blue line actually will also indicate the action potential traveling down the axon. Once it gets down to the axon, there's going to be a trigger releasing all these neurotransmitters into the synapse, right? So postsynaptic neuron is a neuron whose action potentials are propagated away from the synapse. So let's say that enough neurotransmitter is released and enough of these receptors are open such that this postsynaptic neuron will fire an action potential, right? This action potential will then travel down this neuron, right? So that's like the name of the game, right? We're trying to trigger these action potentials to occur, right? And then as we go through, we're gonna find out more and more about what these action potentials are and how they're generated. But for now, let's get like an overall view. And this is probably more important than actually knowing the details. Like if you know, okay, like the purpose of this whole mechanism is so that we can propagate or spread action potentials, then you're pretty much good for now, right? So just know that these neurons are trying to spread the action potentials, trying to spread signals down, right? We're trying to get the signal from the brain all the way down to the hand so that we can contract our fingers, right? So it's a method of signaling. And the postsynaptic neuron, another way to think about it is that this neuron will generate an action potential that will fire away from the synapse, right? So presynaptic neuron generates an action potential that fires towards the synapse. The postsynaptic neuron will generate an action potential that fires away from the synapse. So synaptic knob contains synaptic vesicles. So here is an example of a synaptic knob, right? And this knob contains vesicles that are full of neurotransmitter. Vesicles of these neurotransmitters, right? And these neurotransmitters, you can see them getting released here into the synapse. Yeah, so synaptic knobs have synaptic vesicles. Synaptic cleft, or the synapse, is a space between presynaptic and postsynaptic neurons, right? So just covering that terminology again. So now we're gonna get into like the kind of the meat of chapter 9, chapter 10, because we're going backwards. We're gonna do chapter 10 first, and then we're gonna talk about chapter 9 because it just honestly makes more sense that way. So we have electrical signals, and the purpose of these electrical signals is for cells to communicate with each other, right? So they communicate with each other electrically and chemically. And when you think of electrical signals, they're very brief. So transient just means brief changes in the resting membrane potential. So every neuron has a resting membrane potential. So think of membrane potential as being the same as like the electrical potential or the electrical charge of a neuron, right? So membrane potential is the electrical charge of a neuron, because all cells, I don't know what I was trying to spell there, but electrical, I'm gonna put E in charge, electrical charge. So all cells, for the most part, are inherently charged, meaning that they are negative relative to their environment. So cells are negatively charged, like relative to their environment. And this has to do with a couple of things. It has to do with the fact that the cells themselves, the proteins and everything within the cell are negatively charged. The DNA is negatively charged. So a lot of things within the cell are negatively charged. But also a big part of this is the type of ions that we find in our cells, right? Remember I brought up like sodium and potassium earlier. Those two ions are important in the way that charge is generated within the cell. And we're gonna talk more about that. So I just want to introduce what membrane potential is. Like the membrane refers to like the cell membrane. So membrane potential is like the charge of that cell, like the electrical charge of that cell, whether it's positive or negative. And there's two types of potentials. We have graded potentials, which we're going to talk about first. And again, graded potentials are transient, meaning that they're very brief, and they travel short distances. Right? So these charges will travel very short distances. And then we have the Ash potential, which we'll talk about after we talk about graded potentials. And Ash potentials are electrical signals that travel long distances. Right? Self-replicating means that they will regenerate themselves as they travel down the axon, because Ash potentials will travel down the axon. So these Ash potentials, as they travel through the axon, they will restart or self-replicate at certain points of the axon, so that the signal that travels down is just as strong as the signal that we start off with. And again, like this will make more sense as we kind of go through and kind of fill in the blanks. So again, there's like a lot more terminology. So what early electrophysiologists have done. So what electrophysiologists have done, it's kind of like these are studies that were done like in, I don't know, probably like 50s. They measured the charge within a cell, right? So you have electrodes here. You have a recording electrode, and then you have what we call a ground electrode or a reference electrode. And you have the cell in a saline bath. And when they hooked up these electrodes, what they found was that the cell was negatively charged relative to its external environment. So the saline bath is supposed to resemble the extracellular space that a cell is found in physiology. So they recorded the voltage, and it turns out that a cell at rest has negative 70 millivolts, right? So a cell at rest is negatively charged. That's what it's showing here. And so we're going to look at these graphs a lot. So the purpose of this slide is to show you how we generate those graphs. And how we generate those graphs is we have two electrodes, one inside the cell and one outside the cell. And we are referencing this electrode here. Right? We're comparing the internal environment of the cell to its external environments. And it turns out that the internal environment of the cell is more negative than the external environment of that cell. But as you can see from this figure, this isn't always the case, right? It can always change, right? It can become even more negative, or it can become positive, right? So when the resting membrane potential, is basically the cell is at rest, it's not being stimulated by anything, right? The neuron is not being stimulated by anything. So at rest, it's at minus 70. But this can change when positive charges flow into the cell, right? So when positive ions flow into the cell, that can change the voltage of that cell, right? It can change the membrane potential of that cell. So for example, when sodium is allowed to flow into the cell through those sodium channels, that's going to depolarize the cell, right? And depolarize just means that it makes the membrane potential less negative, or you can think about it as being more positive. You can see here that the membrane potential of the cell increases, right? It becomes more and more positive. So this is when sodium channels are open. Right? Remember, channels, for the most part, sometimes they're open, sometimes they're not open, right? It depends on the stimulus. So sodium channels, when they're open, those sodium ions can flow into the cell, and positive charges flowing into the cell will make that cell more positive, right? So you have to remember that sodium is high outside, potassium is high inside, right? Remember that concept that we talked about earlier? Sodium is high outside, and potassium is high inside the cell, right? Potassium is high inside the cell. Right, so always keep that in mind, right? Because if you think about it, if sodium is high outside the cell and I open up more sodium channels, what is going to happen to the flow of sodium, right? Sodium is obviously going to flow into the cell, right? It's going to want to flow into the cell because of facilitated diffusion, and that's going to make the cell more positive, right? So let's talk about repolarization. So repolarization is kind of the opposite. So repolarization is kind of the opposite of depolarization. So repolarization is when we're trying to return to resting, right? Because the cell always has to return back to its native state, right? You have a neuron, it gets stimulated. After it's done being stimulated, it has to return back to its original state, right? We have to get back to minus 70, right? It's not good for a neuron to be constantly at a positive voltage. So we have repolarization, which is the return to resting after depolarization. So keep this in mind, repolarization can only occur after depolarization, right? Depolarization is positive changes to the membrane potential. Repolarization is negative changes to the membrane potential. They're opposites. And sometimes our membrane potential can become even lower than the resting. So you can see here that this is way lower than the resting membrane potential, right? And we have a special term for this. We call it hyperpolarization, right? Hyperpolarization. The membrane is more negative than the original resting potential. You can see here. So just to reiterate, neurons are negatively charged relative to their external environment, right? In physiology, their external environment is going to be extracellular fluid. And the reason why they're negatively charged is because at rest, there are more potassium channels open than sodium channels, right? So at rest, if you think about it slowly, if you think about it, at rest, there's more potassium channels open. And where is potassium going, right? Potassium, again, you have your cell. Potassium is high in the cell, right? And potassium will always want to diffuse out, right? Potassium, since it's high inside, H for high, and then low outside, the potassium will always want to flow out, right? Because of facilitated diffusion. So just at rest, we just have way more potassium channels open, right? And when potassium leaves the cell, it makes the cell more negative, right? So that should be something that is intuitive. When potassium leaves the cell, when positive charges leave the cell, it makes the cell more negative, right? When positive charges enter the cell, it makes the cell more positive. So that's something that you always have to keep in mind. Like, where are these charges going, right? And the flow of charges is what we call currents, right? The flow of ions or the flow of these charges is what we call the currents, right? So again, I want to reiterate, sodium flowing into the cell will make the cell more positive, and that's what we call depolarization. Potassium flowing out of a cell makes the membrane potential more negative, right? Because you have all these positive charges leaving the cell, right? These are positive charges leaving the cell, and that's going to make the membrane potential negative, and we call that repolarization or hyperpolarization. Hyperpolarization is only if the membrane potential drops below the resting membrane potential, right? So just like take a while to kind of take this all in, because we're going to build on this, right? So now the things, we covered like the basics already. So we're going to talk about like how these changes happen, right? What causes channels to be open, what causes channels to be closed. But as long as you realize that, okay, like where are these ions going? Right? Where are these ions flowing? Sorry, go ahead.
Sorry. So like if a cell is hyperpolarized, would gaining those sodium ions back into it, is that depolarization making it more negative, or is that repolarization putting it back to its homeostasis?
Yeah, so that's actually a really good question. So you talk about how we have hyperpolarization, right? Where we have the resting membrane potential kind of drops, right? It becomes more and more negative.
Yeah.
And the question is, how do we get back up here? How do we get back to resting from a hyperpolarized state? And I think what you said is completely correct, right? So think about what is going to help us make this membrane potential more positive. The only way that the membrane potential can be made more positive is if we have an increase in sodium ions coming into the cell, right?
Yeah.
So that's how we return back to the resting membrane potential, right? Think about what ion do I need to increase the membrane potential? The only ion that does that is sodium, right? So sodium can help me increase my resting membrane potential. Think about also like the opposite of that, right? If I'm at a depolarized state, what's going to help me come back down, right? What event is going to help me come back down? And that's going to be potassium leaving.
Potassium leaving.
Right.
Yeah.
Yeah.
Yeah, so that would still count as repolarization from the hyperpolarized state.
Well, repolarization we only typically use whenever we go down in the membrane potential. So right here, it's kind of like, this doesn't really have a name. But I think some textbooks might call it repolarization. But for the most part, we're just gonna say that it returns back to resting potential. But yeah, so that's like a nuance. I think that changes depending on like what textbook you look at. I think your textbook doesn't really mention. I don't think it mentions like a specific name for this part. But I know that when it goes from positive to negative, it's repolarization. But for the most part, we just call this phase the return to resting potential.
Okay.
Yeah, that was a good question.
There you go.
And so here's kind of all the definitions for you guys in case you want to go back and review them. So let's talk more about these electrical signals, right? So we kind of learned these electrical signals are produced by changes in ion movements, right? Ion movement, again, it's called currents, right? The ion movement is just currents, right? When ions flow from one location to another location, we just call it like currents. And for the most part, this is what determines the membrane potential or the electrical charge of a cell, right? These currents flowing into or out of the cell, right? And there's only two ions that we're really concerned with, just to make things more simple. We have the sodium and the potassium, right? One will flow into the cell and the other will flow out of the cell when given the chance to. And so the big part of like understanding how this works is like what causes them to flow in, what causes them to flow out, what causes sodium channels to open, what causes potassium channels to open, and what helps us get back to our resting potential after an action potential, right? So that's like the rest of like chapter 10's discussion. So again, we have these events, right? These are all triggering events. There's going to be a change in the electric field. For the most part, we're not going to concern ourselves with this. So don't worry about this particular bullet points. But if we think about what is going to cause a channel to open, chemical messengers can bind to a receptor and cause it to open, right? And chemical messengers, again, those are going to be your neurotransmitters, right? Those neurotransmitters can bind to a receptor or a channel and cause that channel to open. And we're going to look at examples of these channels in a bit. But that's one of the ways in which we can get these channels to open, right? The more channels that we get open, the more flow of ions that we have, right? If we shut those channels, then we get less flow, right? So these channels are kind of like the gateways into or out of the neuron. And then here, stimuli such as sound waves, this stuff here, we're going to talk about when we talk about the anatomy of the ear. But for now, we're not going to concern ourselves too much with it. It's just like FYI, keep this in the back of your mind. There's different types of triggering events. There's like chemical events, but there's also physical events, like physical distortions of the cell shape can also trigger channels to be open. But again, this is a concept that we will cover when we learn about the auditory system. And then this one here, don't concern yourselves with this. We're going to keep this simple today, and we're going to talk about mostly this effect here, the chemical messenger effect. And again, the membrane permeability, right? So you're going to hear this word a lot from permeability. Permeability just means how many ions are coming into or out of the cell, right? If we say that the cell is more permeable to potassium, we just mean that there's more potassium channels open, right? So permeate kind of sounds like it's seeping in, seeping out, right? So when we increase our permeability, we're essentially increasing ion flow, right? So that's all it is. Ion flow, permeability, they're all the same thing, right? When you hear something mentioned, oh, there's an increase in potassium permeability, it just means that there's more potassium channels open. If you hear something like, oh, there's a decrease in sodium permeability, that just means that on the neuron, there are sodium channels closing, right? And those sodium channels that are closing, that means that there are fewer ions entering the cell, right? So permeability is just correlated to the amount of channels that are open. High permeability means high amount of channels. Low permeability means low amount of channels open. So here are the first two channels that we're going to discuss. One is called the ligand-gated channel. So a ligand is any type of signaling molecule, and for the synapse, again, the signaling molecule is going to be a neurotransmitter, right? The neurotransmitter will be our ligand. The neurotransmitter will bind to the channel protein, as you can see here, ion channel protein, and it's going to cause this ion channel protein to open, right? And once it's open, these ions that are on the outside can then flow into the cell, right? And when they flow into the cell, that's going to change the concentration of that ion in the cell, and that's going to trigger cellular responses, right? So that's the first type of channel. And I want to point out where these channels are. These ligand-gated channels are going to be located on the dendrites of your neurons. These ligand-gated channels can be located on the dendrites. So on these dendrites, there's a bunch of these ligand-gated channels, right? And again, realize what they do. They're going to bind to neurotransmitter, and once they bind to that neurotransmitter, they're going to open up, and those ions that are in the extracellular space or extracellular fluid, these ions are going to flow into the cell, and that's going to trigger some type of cellular response, right? But of course, they don't remain open forever, because that'd be very bad, right? Because we talked about how this depolarization has to be followed up with repolarization, right? We can't have a neuron remain in a positively charged state forever, right? So that's why eventually the channel will shut and the neurotransmitter will be dislodged from the receptor. Right, so that's the first type. So this one is found exclusively here on these ligand-gated channels, right, of the neuron. So again, you want to imagine that, okay, you have one neuron here, but you also have neurons here. You have a neuron here. You know, you have all these other neurons surrounding this one neuron. And all these neurons are going to be releasing neurotransmitters, right? And these neurotransmitters are going to be binding to the ligand-gated channels of this neuron. And that's going to cause those channels to open so that ions can flow into the cell, right? So that sodium ions in particular will be able to flow inside the cell, right? And another way to phrase this is when the ligand binds or when the neurotransmitter or the ligand binds to the ligand-gated channel, this is going to increase sodium permeability, right? That's going to increase sodium permeability because this channel is open, so sodium can now flow into the cell, right? The other type of channel that's also very important is the voltage-gated channel, right? So the name itself kind of gives away what triggers it to open. Any change in voltage will trigger the opening of these voltage-gated channels, right? So these voltage-gated, so gated means that it's only open under certain circumstances. Voltage-gated channels are only open when there's a change in voltage. Ligand-gated channels are only open when the ligand is bound to that receptor, right? So you can see here's an example of a voltage-gated sodium channel, right? When there's a change in voltage, this guy will open up, and that will allow all the sodiums that are on the outside to come into the cell, right? And so again, you want to think, okay, sodium is coming into the cell. Think about your permeability. If you have all these voltage-gated sodium channels opening, the permeability of sodium increases, right? So it's always proportional. Whenever channels open, permeability will increase for that ion, right? So sodium is flowing into the cell. If sodium is flowing into the cell, what happens to the charge of that cell, right? Was it going to become more negative or more positive kind of thing, right? And again, sodium is a positively charged ion, right? So if you have a bunch of positive charges entering the cell, that's going to make the cell more positive, right? And another word for that is depolarization, right? The membrane potential becomes less negative, also known as more positive, right? And so these voltage-gated channels, they're going to be located here at the neck of the neuron, also called the axon hillock, also called the triggering zone. So all those things, they kind of refer to the same thing. It's like this part of the axon here, the very start of it. There's a high density of these voltage-gated channels. And as we go down the axon, you'll find these voltage-gated channels kind of spread out. Right? So these voltage-gated channels are found all throughout the axon, right? And again, the purpose of these channels is to allow sodium to enter, and so that we can generate an action potential, and an action potential can then travel down the axon and then trigger the release of neurotransmitters here. All right, but more on that later. We're just gonna cover the first part right now, like what is a graded potential? Right, so what is a graded potential? Graded potentials are very small excitations of the cell membrane. And the thing about graded potentials is that they can either be depolarizing or hyperpolarizing, right? Because again, it goes back to the fact that we have different neurotransmitters, right? So here, this neurotransmitter allows sodium to enter the cell, right? So for example, this could be acetylcholine. So acetylcholine is the premier neurotransmitter that will excite the muscle cell. But there's also other neurotransmitters, again, like GABA that we mentioned. GABA can open up a different type of channel that can let potassium out of the cell. Right, so for example, imagine you have another channel here. GABA can bind, right, to this channel, and once the channel opens, potassium can then leave the cell. Right, when potassium leaves the cell, what happens to the charge of the cell? Right, the cell becomes more negative, right, because you have all these positive charges leaving the cell. That's why some graded potentials can be hyperpolarizing, meaning that when these channels are open, the potassium can leave, and when potassium leaves, the membrane potential is more negative. Right? So the thing about graded potentials as well is that they vary in amplitude and duration, meaning that the more stimulation you have, the greater the amplitude, right? So if you have 3 neurons releasing neurotransmitter, right, that's going to produce a pretty strong graded potential. But if you only had one, so for example, if we got rid of these two other neurons, we only have one neuron releasing neurotransmitter, it's not going to have a strong effect, right? The graded potential is going to be weaker. So the graded potential can scale according to how much stimulation there is, right? If there's like 10 neurons around this one neuron, then there's going to be more stimulation, and there's going to be a bigger graded potential. And so this graded potential is produced by various stimuli. We're going to focus more so on the chemical stimuli, right? The neurotransmitter is going to give us a graded potential. And the flow of current is bidirectional, right? So when the sodium comes in, it comes in both directions. It kind of spreads itself out, right? As it comes in, it kind of spreads out. You know, it scatters, it diffuses all throughout the cell, right? And the thing about graded potentials is that they're not very strong, right? So the charges that come in, but they then will dissipate, right? They come in and then they kind of diffuse, and then the amplitude is lost, right? So they come in, they diffuse, you know, and that's it, right? They're not very strong. So I want you to keep this in mind when we contrast this against an action potential, right? So the reason why we discuss graded potentials is because you need multiple graded potentials to generate an action potential, right? So you need multiple graded potentials. In other words, you need a lot of stimulation from other neurons in order to generate an action potential, right? So I'm gonna say that again. You need to understand graded potentials because more stimulation will lead to more graded potentials, and that's going to lead to an action potential, right? More stimulation, more graded potentials, action potential. So you can see that here. The bigger, like, this is showing you the size of different graded potentials, right? And for example, if I just apply one stimulus, this is my graded potential, right? But if I apply even more stimuli, that's gonna be the size of my graded potential. But if I, you know, really amp it up, that's gonna give me a much bigger graded potential, right? Than if I had a smaller stimulus.
Yeah. Kind of like you do all your homework, you get good grades, and you graduate. If you don't
Yeah,
do
yeah,
homework,
yeah. You can think of it that way, right? You can make it hard for
yeah.
yourself and like, kind of like fail classes and like
Oh,
not have that moment. It's like it's a matter of like building momentum, right?
yes.
So it's like how people, how certain people go about the day. They like doing small little things so that it kind of prepares them to do like bigger things. So for example, like you make your bed and you make breakfast, and that's supposed to like ramp up and like eventually you're able to like get your studying done, takes
Yeah,
off from
put
there.
those chores down, then you can move on to other things.
Right. Exactly. And the purpose of this is again to move on to that action potential, right? So we're trying to get to that action potential.
I love life examples.
Yeah, exactly. And so just to reiterate what we were talking about before. So here you have a cell, right? It's at resting. It's at minus 70 millivolts. This is the outside of that cell. It's the extracellular fluid. And you can see from this extracellular fluid, you have all these sodium ions here kind of waiting to get inside the cell, right? But they can't because these channels are closed, right? They're just chilling outside. They cannot enter because the gates are closed, right? It's like lining up before a concert starts or something. So these channels here, again, are your ligand-gated channels, right? You'll often hear them being referred to as chemically gated channels because they respond to neurotransmitters, right? So the neurotransmitter, imagine a neurotransmitter is released, right? The neurotransmitter will bind to that ligand-gated channel, right? You can see that here. And as soon as that happens, the ligand-gated channels will open, and this is going to allow a flow of ions into the cell, right? And why does it go into the cell? It's because of the concentration gradients, right? There's more here than there is inside. So these ions are going to want to go inside because there's fewer ions here, right? So they're going to come inside, they're going to come down the concentration gradients, to come into the cell, right? So again, this is sodium. And when sodium flows into the cell, observe what happens to the resting membrane potential, where it becomes more positive, right? So it's minus 65 as opposed to minus 70, right? And the other thing I want to point out is that it's not always going to become more positive, right? There are certain channels that are opened by different neurotransmitters like GABA that has the opposite effect. It can be hyperpolarizing. These channels can let out potassium as opposed to let in sodium, right? So that's just another example of what you might encounter. But for the most part, we're gonna focus a lot on like these sodium channels, because these sodium channels will allow us to generate an action potential that will eventually allow us to contract our muscle. So that's why we're talking primarily about sodium. So sodium comes in, you have this positive charge coming in, that's gonna raise the membrane potential or make it more positive. So you can see here, movement of ions to the channel produces is local currents. So again, current is just a flow of ions. This depolarizes the nearby regions of the cell membrane, right? So here, imagine that we started off here. Imagine you started off here, right? These ligand-gated channels are opened by the neurotransmitter. You can see that these ions are coming into the cell. As these ions come in, there's current flow, right? These ions don't just stay in one place. They always float around. So some of the ions that come in, they're going to flow to the left. And then some of the ions that come in, they're going to flow to the right, right? So they're going to come in, they're going to flow in different directions. And these positive charges will have an effect on the area that they're located in. So for example, like you measure the voltage here, it's going to be minus 60, right? So recall that we started off at minus 70, right? And so here you have the same phenomenon occurring where you have this channel open and then you have these positive charges coming in. These positive charges spill in both directions, right? And so eventually these positive charges, they're going to get to this location and they're going to change this membrane potential into something that is more positive, right? So as the ions flow, as the current flows, the current will make that area of the membrane more positive, right? And these graded potentials can add up. It's also called summation. They can sum up, and that can lead to an action potential, right? And you might be thinking, okay, like how does it lead to an action potential? The membrane potential of the cell has to be raised to a certain number before it will fire an action potential, right? So for example, the cutoff, which we're going to talk about, it's also referred to as the threshold, is, depending on the book, it's like -50, -55, right? So when you get to this magic number, right, when you get enough positive charges inside the cell and the memory potential becomes -50 or -55, then you get this action potential. And this action potential is much stronger than these graded potentials. And these action potentials are the ones that will actually travel down the axon. Right, so here it's kind of like it's kind of seeing how we're doing kind of thing. So I'm gonna leave this open for a little bit. If you're watching at home, you can kind of also do this. I'm gonna leave it open for like 20 seconds. Right. So hopefully this is kind of clear. Yeah, so depolarizes. So hopefully this is kind of clear because some of these questions are going to appear on the quiz, right? So it's important to understand what happens when positive charges enter, right? Then we have depolarization. What happens when positive charges leave? Then we have repolarization, right? Or hyperpolarization. If there's a lot of charges leaving, our resting or our memory potential can dip below the resting memory potential. Right. So repolarization, positive charges leaving. So again, here is kind of to help you with those previous questions, right? You always want to, the first thing you have to know is you have to know that sodium's high outside, potassium is high inside, right? That's just a fundamental fact that you should know when studying physiology. You should know that there's more sodium outside and there's less potassium outside. And so here's like a summarization of what we talked about, right? These graded potentials and their purpose. And so here you notice this dashed green line here. This is supposed to represent something called threshold potential. When we raise the membrane potential of the cell high enough to this threshold potential, then we're going to have an action potential occur, right? And you can see the action potential has a bigger amplitude than a graded potential, right? So the name of the game is to get to this threshold potential somehow. And for the most part, the only way to get to this threshold potential is to have a lot of graded potentials that kind of add up, right? They have to add up and they have to get us to this -50 threshold. And once you're at that threshold, then you can get an action potential. This is also called an all or none system. If your memory potential is below -50, then you're not going to have any action potential, right? It's just not going to happen. You're not going to have an action potential. But when your memory potential gets to -50 or even higher than -50, then you'll automatically generate an action potential, right? And these action potentials are important because these action potentials will trigger the release of neurotransmitter, right, from the neuron. And so here's an example of an excitatory synapse, right? An excitatory synapse can include neurotransmitters like glutamate or aspartate, so you don't have to know these. The only neurotransmitter that's a requirement for these next two chapters, or for like 9 and 10, is acetylcholine. Okay, so acetylcholine is a positive, or it's an excitatory neurotransmitter. Right? The reason why is because acetylcholine, we're going to talk about it a lot when we talk about the muscle later. Inhibitory synapses, just basically for FYI, it's not always going to be excitatory. Some neurotransmitters are actually inhibitory, meaning that they're going to take us farther away from threshold, right? So we see hyperpolarization here. We're farther away from threshold, right? Because this is original, and then this is what happens when we have an inhibitory synapse. We are farther away from the threshold, meaning that it's going to be harder for that cell to generate an action potential. Right? It's going to be harder because you're working from here. You're working in a deficit, right? You're like working here, and you're trying to get to this threshold here. Whereas here, it's different because you have all these excitatory neurotransmitters that are just increasing the memory potential. And eventually, that's going to allow us to get to threshold, right? So Again, threshold is going to be this magic number minus 50. When you get that number, you're going to fire an action potential, and that's that, right? If you're below that number, if you're like minus 51, you're not going to fire an action potential, right? You have to be like minus 50 or higher. It's kind of like how grading systems work, right? If you, in order to get an A, you can either get like a 98% or like a 95%, right? Like either one is going to give you an A in the class, right? It's kind of like that. And so here, kind of to backtrack and reiterate some important points, you have the input zone, and recall that this is where your ligand gated channels are going to be, right? And then you have the trigger zone. So here you have your voltage gated channels, right? Voltage gated channels here at the trigger zone. And then you have voltage gated channels like all along this axon, right? The axon itself is called the conducting zone, So the action potentials that travel down are undiminished, meaning that they retain their original voltage as they travel down, their original strength, if you call it. And this is important, right? Because we want that signal strength to maintain, to be strong as it travels down the axon, right? We don't want it to get weaker, right? If it gets too weak, then nothing is going to get here, right? There's going to be very little signal here. The whole point is to get the signal to travel from here all the way to here, right? Try to get all the way to the very, very end, right? That's the whole point. And that's why the action potential has to be self-regenerating. So it has to regenerate itself at different points in the axon so that it maintains the same signal strength as it travels down the axon. If for some reason there's like a break here and current flows out, then you're gonna lose that signal strength, right? And you're getting to this point where there's like barely any signal, right? It's kind of like your cell phone carrier, right? Like if the signal is not strong enough, then you're not gonna get your call.
Reminds me of a running car. It charges itself as it's running.
Yeah, right. Exactly. Kind of, yeah, it's definitely kind of like that. But yeah, for the most part, yes. It will regenerate itself as it's kind of traveling down the axon, yeah. That's a good comparison. So yeah, so now we're going to transition over talking about action potentials, right? So action potentials, here are some characteristics. They are very rapid. They're large changes, large electrical changes in the neuron, right? And we're talking about like 100 millivolts. And the thing is, it only involves a small part of the excitable membrane. It's the part here of the axon, right? And the action potential will regenerate itself at very small parts of the membrane, as we'll soon talk about. So non-decremental, meaning that it maintains its strength as it travels down the axon. One-way propagation just means that the action potential will only travel in one direction. It's not going to backtrack and go back up here, right? That's not going to happen. That is something that graded potentials do, but action potentials are unidirectional. They only travel from here all the way down, right? So all or none phenomenon, meaning that you need a threshold potential of like minus 50, right? Remember, minus 50, it's all or none. If you're at minus 50, then you're good to go. If you're at minus 60, there's not going to be an action potential, right? So all or none. Stereotypical size and shape means that whenever you generate an action potential, it's always going to be the same size and shape. The magnitude or the strength of that action potential will always be the same. It doesn't matter, what the initial triggering stimulus is, whether it be like minus 50 or minus 40, right? It doesn't matter. The action potential that you generate will always be the same size and the same shape, right? So other names for it that you will probably encounter are spike, firing, right? That's what we call action potentials. And here's an action potential, right? So if we look at the kind of the anatomy of an action potential, here we start off at resting, right? These are all graded potentials that are summated, they're added together, right? Summation of the postsynaptic potentials, right? PSPs are postsynaptic potentials or graded potentials, GPs. And once you get high enough, once you hit that threshold, right? This guy here, then you will fire an ash potential, right? So the red, this is all ash potential, and then you get back to resting, right? So again, one way, non-decremental propagation, all or none. So let's go back into and kind of talk about the main players in this, the generation of the ash potential. There's for an ash potential, there's like specific channels involved, and we mentioned these already. These are the voltage gated channels, right? You have the sodium voltage gated channel, and you also have the potassium voltage gated channel. And these voltage gated channels are triggered to respond at threshold, meaning that when you get to threshold, that's when the voltage gated sodium channels open, and that's when the voltage gated potassium channels begin to open. So voltage gated channels, they will only respond when you're at threshold. So when you're at threshold, the voltage gated channels will respond, right? So let's take a look at these different types of channels. So the first channel that we're looking at is a voltage gated sodium channel. And the thing about the voltage gated sodium channel is that there's three states. There's a resting state, there's an activated state, and an inactivated state. So the resting state, as you can see here, this guy shuts, right? This sodium ion is trying to enter the cell, right? Just so that we orient ourselves. This is the extracellular space. This is intracellular. And the sodium, remember, sodium is high outside, low inside, right? High sodium outside, low sodium inside, right? So guess where the sodium wants to go? It's gonna wanna go inside, right? But it can't because this channel is shut. But, you know, this occurs at -70, so this is at resting, right? At resting, these channels are shut. But when you get to -50, when you get to threshold, this voltage gated sodium channel will open, right? And when it opens, right, just like with the ligand gated channel, the sodium is gonna wanna flow inside, right? And think about the repercussions of this. If sodium comes into the cell, is the cell gonna be more negative or more positive, right? You should think about that. So as sodium comes into the cell, it's gonna make the cell more positively charged, right? And this continues to occur until the peak. The peak is gonna be plus 30 millivolts, right? Plus 30 millivolts is gonna be the peak of our ash potential. And again, like depending on the book, they always, you know, the number kind of fluctuates a little bit, but you know, plus 30 is a good number, kind of a good mean. So this is going to be the peak of our ash potential. Once it gets to the peak, something interesting happens. It does not close, right? It doesn't go back to its resting state. It transitions to a different state called the inactivated state, where you have this part of the protein that will slide in and close the channel that way, right? So you call this slow closing, and this is triggered again at the threshold, and it doesn't really fully close until we get to the peak ash potential, right? And so this is interesting because even though this part is open, right, this part is closed. So sodium will still not be able to enter the cell, right? Just because this part of it is closed, so no sodium can really enter it. It's inactivated, right? And it's gonna stay inactivated until we return to resting at -70, right? So we think about permeability. Which three of these examples is sodium permeability highest, right? It's gonna have to be this one. So sodium permeability is highest when this voltage gated sodium channel is open. Sodium permeability is low when the channel is in the resting state or the inactivated state, right? So now let's talk about the voltage gated potassium channel. So again, you have this activation gate, right, like you did for the sodium channel, but there are differences. So for the voltage gated potassium channel, it remains in this state from resting all the way to +30, right? So this guy opens a lot later than the voltage gated sodium channel, right? This guy opens at +30. So at +30, he opens, right? And at +30, this guy is inactivated. And so no sodium's coming in. At the +30 mark, only potassium is leaving the cell. And there's no sodium entering the cell, only potassium is leaving the cell, right? So this guy opens, and then we have the potassium ions leaking out of the cell, right? So think about what the repercussions are of this. If potassium is leaving the cell, that's gonna make the cell more and more negative, right? And this is the repolarization phase of our action potential. Right? This is the repolarization phase of our action potential. And so here it kind of summarizes, I think, everything that we've mentioned thus far. So I'm gonna leave this for you guys to read just as a reminder. And we're gonna take a look at this figure here that kind of sums everything up very, very nicely. And so here you have your action potential, and then here it's gonna show you what happens at each junction of that action potential. So if we're at resting, right, which is here at 1, at resting, look at the channels. At resting, our voltage gated channels are both closed, right? They're both closed, right? But when we reach this threshold, because of the graded potential, so imagine you have some kind of graded potential, and imagine that we eventually get up to the threshold potential, that at this point, that's when these voltage gated channels are triggered to respond, right? They're triggered to open. The thing is that the voltage gated sodium channels will open first, right? The voltage gated sodium channels open first. Remember, it takes a while for the voltage gated potassium channels to open. They have what we call delayed opening. So when you have a bunch of these voltage gated channels opening, right, what is happening? What is happening? These sodium ions are coming into the cell. And what are these sodium ions doing? They're making the cell more positive. They're going to make the cell more positive. So more positive. So that's why you see here sodium in. This is the rising phase, right? We have depolarization due to the massive influx of sodium. So at this point here, sodium permeability decreases, potassium permeability increases. Because at this point here, the sodium channel, the voltage gated sodium channel is inactivated, right? So that means that no more sodium is coming in. The only channel open here is going to be the voltage gated potassium channels. And they're finally going to open, right? Remember, they have delayed opening. So it takes them all the way here before they can actually open up. Once they open up, the potassiums come out, right? Positive charges leaving the cell. Positive charges leaving the cell. That's going to give us our repolarization phase, right? Give us our repolarization phase. Can remember the y-axis is giving you the voltage of the cell, right? We have a decrease in voltage. This is called the falling phase, right? As the potassiums are leaving the cell. Eventually, we actually hyperpolarize. Too many potassiums leave the cell. At around, again, back at around the threshold points, the voltage gated channels are triggered to reset, right? They're triggered to close, right? So here they're triggered to open at the threshold. When they go back to the threshold again, upon the falling phase, they're going to be triggered to reset to their original conditions, right? So then you have hyperpolarization, where the channels are just closing, and then you get the resting again, right? We're back to our resting potential, and you see at resting again, the voltage gated sodium and potassium channels are both going to be shut, right? So the P's here are representing permeability. Right, so the other thing, I think Jody mentioned, like, how do we get back to the resting potential, right? If we always hyperpolarize, like, what's going to bring us back to the resting potential? One of the things that does that is a different kind of protein called the sodium potassium pump, right? This guy should be familiar because he's a primary active transporter, meaning that he uses ATP to pump ions against their concentration gradients. And the other thing that happens is that we have leaky channels. So leaky implies that these channels are just always open, and they're going to allow the ions to flow down their concentration gradients. Okay. So both of these are going to help in resetting that membrane potential, right? You have the sodium potassium pump, which is gonna pump sodium into the ECF, pump potassium into the ICF. So basically pumping both these ions against their concentration gradients. But then you also have the leaky channels that allow charges to come in and come out of the cell, right? And those two things are supposed to help us reset back to our resting potential. Right, so let's think about this question. It's kind of a thought-provoking question. You guys have probably heard of fugu, or this guy here is a pufferfish. If you're like super serious about sushi, you probably had a pufferfish before. These fish are highly toxic because they have this organ that produces a toxin called tetrodotoxin. And tetrodotoxin can actually cause nerve system blockages and loss of sensation, paralysis, and even death in high concentrations. And it happens so fast that if you have fugu that is improperly prepared, you just die immediately. Right? So it's such a potent toxin that it will just kill you immediately if you have a chef that doesn't know what he's doing. Right? If they don't know how to remove the poison sac properly, and a bit of the poison is left within the fish, then that minute dosage is enough to cause respiratory arrest. Right? So you won't be able to breathe. Obviously, you're going to die. But if it's prepared properly, it actually gives you a very pleasant tingling sensation in your mouth. So, never had it before, so I don't intend to. I don't know if you guys ever had it, but feel free to speak about it if you have.
Nope, not a big sushi fan.
Okay, then. FYI, this fish exists. So I'm gonna leave this open for like 30 seconds and give you guys a chance to analyze this question.
So why is a catfish so venomous, so poisonous?
I actually don't know what kind of toxin that I didn't even know catfish was poisonous until now that you mentioned it. I don't know much.
Yeah, it's the same concept. If the sushi preparer doesn't prepare the catfish correctly, it's also poisonous.
That's interesting. Yeah, I don't know. I'm pretty sure it's not. Probably different toxin. Probably different neurotoxin.
Yeah, well, I'm thinking maybe the whiskers has something to do with it.
Yeah. But those guys are like bottom feeders, though.
Yeah.
They're like, you don't want to eat bottom feeder fish.
Yeah. Imagine how many toxins are under there.
Exactly. All right. So let's take a look at this. Okay. So let's observe this curve really quickly. So we mentioned that the tetrodotoxin, how it works is it kind of blocks these voltage-gated sodium channels, right? So basically, it prevents them from opening. So what part of the action potential graph is affected? So we go back to our figure here, and it looks like at this point, right, we have opening of the voltage-gated sodium channels. And so if you have the toxin come in, the toxin will actually prevent this from opening, right? And that is going to affect probably like this part of that curve, right? So you're not going to see this peak at all, right? Because what generates this peak at C, that's going to be the voltage-gated sodium channel, right? That's going to give us our big depolarization, right? And then think about what is responsible for all the events that are happening here. So at B, this is the graded potentials being caused by the ligand-gated channel, right? And then once we get to threshold, you have this big spike here, and again, this big spike is the voltage-gated sodium channel. Like all those positive ions coming into the cell, making that cell more positive. And then you get to this point here where the voltage-gated sodium channels, they will inactivate, and the voltage-gated potassium channels finally open. So the point at D, when we start to repolarize, that's going to be due to the voltage-gated potassium channels, right? So each segment of this exponential curve can be explained by what channel is active at what time, right? So again, to reiterate, to get us to threshold, that's going to be primarily due to the ligand-gated channels on the dendrites of our neurons. Once we get to action potentials, the voltage-gated sodium channels take over, and then you have this massive influx of sodium. It's going to bring us all the way to the top. So once we get to the top, then we have the voltage-gated potassium channels opening, the voltage-gated sodium channels inactivating. They're going to be the only channels that are open. So the potassium is going to flow out of the cell. And think about what happens when positive charges leave the cell becomes more negative, right? Eventually, we will hyperpolarize and then return back to our resting phase. And so here's another question, and it deals with a different type of toxin. I always butcher this name whenever I read it. I think it's like batrachotoxin, and you can find these on the skin of dart frogs. And you can actually, like, what they'll do is that the hunters will coat their arrows. They like rub the arrows on the surface of the skin of the frog, and it will poison the tip of the arrow so that whatever the arrow touches will result in paralysis, like muscle paralysis. So how does this toxin work? It works by irreversibly opening this voltage-gated sodium channel, right? So different mechanism here, right? So for the the TTX or tetrodotoxin. Tetrodotoxin prevents them from opening, whereas this toxin here, okay, well, I'll kind of read the answer, but this toxin here will keep them open forever, right? So they will open, and so if we look at D, right, the voltage gated channels, sodium channels are going to open, but they do not shut. They remain open. They remain open. And too much sodium enters the cell, and this is going to prolong the life of your action potential, right? Because recall that the x-axis is time. So the action potential will persist for a longer period of time, right? And eventually, this is again, like FYI, but eventually what will happen is that all your voltage-gated sodium channels are going to be activated, right? And what follows activation? What follows opening, right? It's going to be inactivation, right? When they open, they're going to inactivate. So you're going to have a bunch of these voltage-gated sodium channels just inactivating. And since they're all inactivated, that's going to lead to flaccid paralysis. So paralysis where there's no stimulation, there's no more output for the neuron. There's like different types of paralysis, where there's like spastic paralysis where your muscle just like contracts rapidly, kind of like muscle spasms or muscle cramps. Like sometimes, I don't know if you guys experience like running, like for some reason, like your calf muscle or your gastroc just kind of like starts spasming and you can't really go, and it's kind of painful. You can't really run anymore. You see this with like Olympic athletes too. Sometimes like quad will be spasming, and it just hurts a lot because it's muscle just rapidly contracting and relaxing. And there's another type of paralysis called like flaccid paralysis, where there's like no input to the muscle whatsoever. And so that's the type of paralysis that this toxin executes. It's just flaccid paralysis. There's no activation of the muscle, right? So you can try to like contract, like the animal that's poisoned can try to like contract their legs if they want to, but it's not going to fire the muscle.
Wouldn't that like
Yeah.
the constant sodium entering the intracellular fluid, wouldn't that also cause your cells to eventually explode if all your extracellular water goes into the cell? You know what I mean?
Yeah, that can definitely be, definitely. Yeah, that can be a possibility for sure. Right. And I like how you tie that concept together, like that osmosis concepts, right? The sodium coming in, the cell will swell up. Definitely. So now let's talk about the refractory period. So, the refractory period kind of talks about how the charges travel down the axon. We talked about how the action potential is self-regenerating, but this is kind of showing you why it's one directional, right? So this refractory period shows you why propagation is one way. Because what will happen is that, imagine as it travels down, right? So this is the area that we're going to focus on, right? This is the new active area. So this area here will fire an action potential, right? And basically what will happen is that you have all these positive ions coming in, you have all these sodiums coming in. And again, ions, they will flow in both directions, right? Because they're going to come in, they're going to diffuse, they're going to spread themselves out. But there's a reason why, like the previous section does not fire an action potential too, right? Only the sections downstream will fire an action potential. The regions upstream will not fire an action potential. The reason for this is because of those voltage-gated ions, voltage-gated sodium channels being inactivated. Right? So there's a purpose to this, and the purpose of the inactivation phase is to prevent a backflow of action potentials, right? So the action potential will not travel backwards because all the voltage-gated sodium channels here are going to be inactivated. But the ones here are still in their closed phase, right? So that means that they can be triggered to their open phase. They can be triggered to their open phase. And so that's why the action potential only goes in one direction. So even though the ions flow in and they spread themselves out and they travel, these ions flow to both directions of the axon, this region has just fired an action potential. So it's not going to be able to fire another one, right? So it's kind of like resting. So refractory just means resting phase. And this ensures that our action potential will only travel in one direction. And you can read about it here, basically what I just mentioned. I don't even know why I went backwards when I had the same figure here, right? Because here you have like the inactivated phase, right? Inactivated phase here. And everything downstream here is in the resting phase. And if it's in the resting phase, that means that it can be triggered to the activated phase, right? And then here, the segment that we're focusing on just fired an action potential. So this would be the activated phase here, right? So they're in 3 distinct phases: inactivated in the back, activated currently when you have the charges coming in, and then downstream you have resting, right? If you're in the resting phase, that means you're ready to be activated. But if you're inactivated, you can't be activated anymore, right? That's why it's called the inactivated phase. So this ensures unidirectional traveling of the action potential. And the last thing about it, the thing that speeds up an action potential is myelination. So myelination, as you can see here, this axon is covered by this layer of fat. Like this is what we call a lipid sheath. And you'll have this in the peripheral nervous system as well as in the central nervous system. So in the peripheral nervous system, they're called Schwann cells, and in the central nervous system, they're called oligodendrocytes, but they're essentially the same thing. They're a lipid sheath that surrounds the axon, and with this comes a lot of benefits. Right? As you can see from this graph, with a myelinated axon, the conduction velocity, so the speed of the action potential, is a lot faster than an unmyelinated axon, right? So myelinated, the signal travels a lot faster, which is ideal. That's what we want. Or we want signals to travel fast. I don't want to wait like 1 second for my bicep to contract when I want it to contract. I want the contraction to happen like almost like simultaneously. As I think about it, I can do it. That's how fast it is, right? So that's why majority of our motor neurons that trigger muscle cell activation, they're going to be myelinated. And another thing that influences speed of conduction is diameter. The bigger the diameter, the faster the signal can travel, just because there's less resistance. But for the most parts, myelination is very important. Right? And myelination, sorry, go ahead.
The myelinated sheet kind of prevents that signal from technically escaping in a way.
Exactly, yeah. So that segues into my next point, right? This sheath here, you can see covering it, it prevents charges from leaking out of the axon. So it kind of traps the charges inside, kind of like how an electrical wire works, right? If you look at an electrical wire, it has like the rubber covering on it. That's to prevent charges from leaking out and like shocking you. So that's the purpose of the axon as well, right? You have this sheath surrounding us. These ions, this current will remain inside the axon.
Okay.
Yeah, that's a good points. Yeah. And the other thing as well that I want to point out is there's gaps between sheaths, right? So you have a sheath here, and then you have a gap, and then you have a sheath here. This gap is called the node of Ranvier. And the node of Ranvier has a high density of these voltage-gated channels, and it's here that the action potential will be regenerated, right? So remember, the action potential has to be regenerated, otherwise it's going to lose its strength as it travels down the axon. It'll get regenerated at the segments here, at all these segments here. And the myelinated sheath will ensure that no charges leak out, so that the signal strength is maintained here as it gets to here, right? As it jumps from node to node, the signal strength is going to be the same. And when the action potential travels down, that's going to cause a voltage change that will activate these voltage-gated sodium-potassium channels, right? And that's going to give us action potentials because these channels open. You can see more ions come in, and that's going to regenerate the strength of our action potential, and we call that saltatory conduction. Salta is like jump, right? It kind of jumps from node to node to node, right? So you can see the comparison here. You have an unmyelinated axon and a myelinated axon. It travels a lot faster, and it also can regenerate at each node, right? Whereas you can see here, I think Jody, you made the points that the sheath is there to insulate the charge, and you're right, because you can see here in this unmyelinated axon, the charges are starting to leak out, right? So you can see there's a decrease in signal strength, right, as it travels down the axon. Whereas here you can see there's no issue for the myelinated axon because we have regeneration at this junction. And so why is this important? Why should things be myelinated? You guys probably heard of multiple sclerosis. Multiple sclerosis is this autoimmune disease where your body attacks these myelin and that's going to cause the sheath to disappear, right? And we all know what happens when the sheath disappears. The signal doesn't get to where it's supposed to go, right? So that's why multiple sclerosis, you lose the ability to contract your muscle, right? You lose the ability to control your skeletal muscle. Right, so we get to the very end, and this is the whole point of the ASH potential. The whole point is to cause the release of this neurotransmitter, right? And I'm not going to cover it because we already covered it. We're basically did like a full circle loop, right? Where we get to the end of the axon, the ASH potential is here, the ASH potential triggers all these things to occur, right? It triggers the opening of the voltage-gated calcium channels. There's like voltage-gated calcium channels that are only found at the terminal. These voltage-gated calcium channels are then going to allow calcium to come in, and calcium is going to allow neurotransmitter to be released into this synaptic space, right? The synaptic cleft. And the neurotransmitter is going to bind to the postsynaptic cell, as you can see here, and it's going to cause the same thing to occur again, right? So we basically made a full circle thing, right? So ASH potential will trigger voltage-gated calcium channels opening. Calcium will trigger these vesicles to release neurotransmitter into the cleft. Neurotransmitter will then bind to the postsynaptic receptors, and this postsynaptic cell will then be stimulated, right? And here's like a little explanation for that, but we basically just mentioned it. And then this slide here is to show you that the neurotransmitter is supposed to be destroyed or brought back to the cell, right? If it stays too long, then it can actually damage this postsynaptic neuron. And since we know about this mechanism, you can actually design drugs that can mess with the synapse, right? So imagine, I'm pretty sure you guys have heard of like antidepressants or SSRIs, right? The purpose of those SSRIs is to maintain a high concentration of serotonin, like in the synaptic cleft. Because what will happen is that, imagine serotonin gets released, but these things happen to it, right? Since this is just like a space, the serotonin can actually like diffuse away from the synapse, right? Instead of binding to its receptor, the serotonin can actually be degraded by enzymes, right? Just random enzymes that will break down the serotonin, or the serotonin can be pumped back to the presynaptic cell so that it can be stored and then used again later. But drugs like SSRIs, they will actually prevent this reuptake, and so that the serotonin remains in the synaptic cleft for longer. And that's going to mean that it's going to have a longer effect, a more prolonged effect on this neuron here. All right, so that's what SSRIs do. Antidepressants, for the most part, they function by keeping the neurotransmitter at the cleft for a longer period of time. And so now we're going to finally talk about muscle. I'm going to transition over the time we have left. And, you know, here's the reason why we spend so much time talking about neurons. Because without neurons, then you will not have voluntary control of your muscle, right? So here's your spinal cord. You have neurons coming out of it, right? This is the motor neuron. Motor neuron will then synapse onto the muscle cell. So you can see that here. This is like a histological section. It's kind of stained. So you can visualize the muscle fibers, and you can see how the axon kind of branches out and innervates the muscle, right? This is what allows our muscle to contract. And one neuron can actually innervate many of these muscle fibers, and we call that whole thing the motor unit. So the motor unit includes the motor neuron that stimulates the muscle, as well as all the muscle fibers that are stimulated by that motor neuron. So some terminology things to kind of cover. So remember we call this space the synapse, right? This is a synapse. For muscle, this area between the neuron and the muscle, we call the end plate potential. So instead of graded potential, we call it end plate potential. It's kind of the same thing. So instead of synapse, we call it the neuromuscular junction. Sorry. The synapse means the same as neuromuscular junction. It's like a bit of a name swap because we're talking about axon on muscle action now. End plate potential, sorry, is just referring to graded potential. All right, we're talking about graded potentials, how they have to build to get to an ASH potential. For muscle, it's called end plate potential, but the concept itself is the same thing. It's just a name change because we're talking about muscles now. Same thing for the synapse. So instead of a synapse, we call it the neuromuscular junction, right? If you think about it, if you break this word down, you got the neuron, and then you got the muscle, right? Neuromuscle junction. right? And again, I mentioned acetylcholine before. It's going to be abbreviated ACH. Acetylcholine is like the main neurotransmitter that is released at the neuromuscular junction. Some other things as well. So the plasma membrane of a muscle cell is called the sarcolemma. So basically sarcolemma is the muscle membrane. Oh, sorry, is the plasma membrane of the cell. So again, name change because we're talking about muscle cells now. But everything else is pretty much the same as the neuronal cell. The other thing to mention is the resting memory potential is a lot lower. It's minus 90. But to be honest, like this is just FYI. Don't worry too much about it. You should just know that the plasma membrane, the name of it is sarcolemma, right? But everything else is the same, right? We have self-regenerating action potentials. Everything else we talked about has carryover here. The motor end plates is what we call the area that is next to the synaptic terminal. This is an area that can be depolarized, but the channels here are chemically gated, right? They're ligand gated. So chemically gated just means the same thing as ligand gated, meaning that they can be opened by a neurotransmitter. And again, when we talk about muscles, the main neurotransmitter is acetylcholine, right? So that's why we call these receptors at the motor end plate acetylcholine receptors, because these receptors bind to acetylcholine. And they're capable of graded potentials, right? Also called end plate potentials, right? So again, same exact concept as before, right? What triggers graded potentials in the neuron cell? That's going to be neurotransmitter release, right? And opening of the ligand-gated channels. Same thing here. What triggers graded potentials? Also known as end plate potentials in the muscle cell. It's going to be the acetylcholine binding to acetylcholine receptors. Right, so, and again, more terminology, synaptic vesicles we talked about already. Active zone is where the vesicles are stored and when they're, so that they can be released rapidly. The motor end plates is, again, we mentioned this already, the sarcolemma that is opposite to the terminals. And this is where you're going to find the receptors for the acetylcholine. Motor end plates is the site of the acetylcholine receptors. And acetylcholine receptors, just to make it more simplistic, you should know that they're going to let in sodium ions, right? So this is like another level of detail that you don't have to know. Just know that acetylcholine receptors, sodium ions, right? Sodium ions are going to come in. And end plate potential is the same thing as a graded potential, right? Whenever you see end plate potential, you should know that we're talking about muscles, and you should know that it means the same thing as a graded potential. And what triggers a graded potential, or what triggers the formation of an end plate potential? That's when the acetylcholine receptors are opened. Right? When they're opened, the cell is depolarized, end plate potential will then combine and eventually form an Nash potential, right? So it's the same exact concept. End plate potentials, once you have enough of them, you're going to lead to the formation of an Nash potential. And the Nash potential will spread all throughout the cell. So let's take a look at this neuromuscular junction. So here you have the motor neuron, right? Motor neuron releases the acetylcholine, right, onto this muscle cell, right? So a couple of things here. You have your voltage-gated channels that are allowed these vesicles to release the acetylcholine. You have this enzyme called acetylcholinesterase just waiting there, and the purpose of this guy is to break and destroy all the acetylcholine. Right? He's chilling here because his job is to kill all the acetylcholines that come out. Right? The acetylcholine will then bind to acetylcholine receptors on the motor end plates. Right? You can see these are all acetylcholine receptors and they're all bound. And once the binding occurs, sodium can then enter the cell and depolarize that cell. Right? Same exact concept as before. Sometimes the acetylcholine receptors are also called nicotinic receptors. The reason why they're called nicotinic receptors is because nicotine can also open the neurotransmitter, but endogenously, so physiologically, acetylcholine is responsible for opening these receptors. But what they found is that they can also stimulate these receptors to open with nicotine. All right, so these acetylcholine receptors are not only on the muscles, but they're also distributed all throughout the brain. So that might be the reason why, like, people who smoke cigarettes, you know, the nicotine nicotine is just binding to their nicotinic receptors in the brain and having leading to, just heightened awareness, all that stuff that comes with nicotine, perhaps addiction. Yeah, so you can find them in the muscle. You can also find them in the brain. They're actually located pretty much everywhere. And here's kind of a summarization of what we're going to talk about. So what happened at the neuromuscular junction? So here you have your motor neuron, right? Somatic motor neuron. Releases acetylcholine. Acetylcholine binds to acetylcholine receptors at the motor end plates. That's going to trigger the formation of end plate potentials, right? You can see the sodium coming in, right? It's going to trigger end plate potentials. When you have enough end plate potentials, that's going to lead to an ash potential. The ash potential will then travel down something called the T-tubule and reach the deeper muscle fibers down below. And the ash potential, the purpose of this ash potential now in the muscle is to open up this guy, right? These two dudes, DHP and RYR. These two channels are these calcium releasing channels that allow calcium to be released into the cytoplasm of the muscle. Because usually calcium is stored in this sarcoplasmic reticula. Right? It's like a storage place for calcium. But when an ash potential comes down, it's going to cause these calcium releasing channels to open. Calcium escapes. Calcium will then allow this to occur. The myosin and actin binding to each other. Right? But for now, we're going to stop here. We're going to focus here on this part, right? So if you read this, then you're going to pretty much understand everything up to this part. And again, this is like a resummarization of what we just talked about before, right? If you wanted a different picture to look at. We're going to talk more about muscle anatomy before we proceed on to what happens like down here and all this stuff here. We have to understand a little bit of the muscle anatomy. So the thing about muscle is that it's very compartmentalized. So you can see that you have this muscle here. The muscle is actually made up of a bunch of fascicles. Fascicle is a bundle of muscle fibers. So here in this one fascicle, you have a bunch of muscle fibers. Here's another fascicle, you have a bunch of muscle fibers. And if we take this out, right, we take out this fascicle and we look at the muscle fiber, each of these muscle fibers have myofibril, right? And this is, again, taking a look at the muscle fiber. If we pull out one of the myofibrils, this is what you're gonna see. And the myofibrils themselves are made up of a bunch of different proteins called myosin and actin. Okay, so if you zoom in on a myofibril, this is what you're going to look at. You're going to look at a sarcomere. And another thing I want to point out is you have the T-tubule here in between these sarcomeres, right? You have the thin filaments called the actin, thick filament also called the myosin. And you can see that this whole thing is surrounded by these T-tubules, right? And these T-tubules are going to allow the ash potential to spread to the deeper muscle fibers. And that ash potential is going to cause the release of calcium from the sarcoplasmic reticula. And the calcium will then float and bind here on a protein called troponin. So I want to show you guys that. Because what will happen eventually is that the myosin and the actin, they're going to interact with each other and they're going to shorten. And so the myosin here is going to bind to the actin and it's going to pull it towards this part of the sarcomere called the M line. All right, so you have the myofibril. If we look at one segment of that myofibril, you'll see that there's a bunch of sarcomeres that make up a myofibril. So it's like kind of inception kind of based where you have if you look at it like this is the muscle. Within a muscle, you have fascicles. Within a fascicle, you have muscle fibers. Within a muscle fiber, you have myofibrils. Within the myofibril, you have sarcomeres. Right? Sarcomeres are like the basic units. And here in the sarcomere, within a sarcomere, you have a myosin and an actin. And the myosin is going to bind to the actin, pull it in towards the M line. And it's going to do the same on this side. And that's why your muscle shortens when you contract it. Right? Have you ever noticed that when you shorten a muscle, to generate force, the muscle looks shorter. Right? It looks shorter. Like you contract your bicep kind of bunches together, it looks a lot shorter. Right? And this length change is what will generate force for us. And so here you have your myosin, also known as the thick filaments, and you have your actin, also known as the thin filaments. And then on the actin, I want you to notice that there's a few proteins here that we're going to be talking about called tropomyosin, troponin. Troponin is going to be the protein that binds to calcium. So again, kind of showing you that the muscle will contract when calcium is present. Right? So calcium will bind the troponin, allowing myosin to interact with the actin. Right? And during a muscle contraction event, the sarcomeres are all going to shorten, as you can see here. Right? So relaxed versus shortened. So every zone will shorten except for this A band here. A band remains the same, but you can see that the I band is going to shorten and the H zone will shorten as well during muscular contraction. All right, so here's like a description of all the different zones on a sarcomere. Because now we're going to dive into the rest of the physiology, right? So we stopped at the action potential parts. What happens at the action potential? What's the purpose of an action potential? It is to depolarize the T tubules, right? So you can see the action potential will travel down the T tubule and cause the release of calcium. Right? That's the whole point of this action potential, cause the release of calcium. Right? So this is just FYI right here. The main takeaway from this is that we have calcium release. Right? So when the action potential travels down to the T tubule, we have calcium release. And of course, it's done by like very specific proteins, and it's even more complicated mechanism, but don't worry about this part. This is FYI if you want more information. So what is calcium? What is the purpose of calcium? Why are we trying to release this calcium? What does it do? Calcium is important because it binds to a protein called troponin, and that's going to allow the myosin to interact with the actin. That's going to allow our muscle contraction to occur. Right, so here's a description of the myosin and actin. And then troponin and tropomyosin are proteins that you'll find on the actin. So TN is troponin. Troponin binds to calcium when it's present, and that's going to cause the tropomyosin to shift, exposing the binding site on actin. Right? So when the binding site is exposed on the actin, that's when the myosin can bind to it, and then that is going to cause a power stroke and a shortening of the sarcomere. So you can compare and contrast that here. In the relaxed state of the muscle, right? There's no calcium, right? Relaxed state, no calcium. That means that the myosin cannot bind the troponin. So in the contractile state, when the muscle is ready to contract, you can see that there's calcium present, right? Because of the action potential. The calcium will then bind the troponin. This is gonna expose the binding sites on the actin so that the myosin can bind to it. And then that's gonna shorten our muscle, right? And all of this it's a part of something called the sliding filament theory or the cross-bridge cycle. And so we start here at the very, well, it's a cycle, so you can basically start anywhere. But here we are right after muscle contraction. So right after a muscle contraction event, the myosin head is attached to the actin. So what needs to happen is that there needs to be detachment of the myosin head. So ATP comes in, it binds to the myosin head, causing it to detach from the actin. You can see that ATP is broken down into ADP and phosphate. It's hydrolyzed by the myosin, so hydrolyzed just means breaking down. So the myosin has a protein that will break down the ATP, and it will take that energy from the breakdown and enter what we call the cocked state, so the energized state. Right? So now it's ready to bind to the actin, but it cannot. Right? It cannot. And the reason why is because there's no calcium here, right? So even though the myosin is ready to go, the binding site for it on the actin is still blocked by that tropomyosin protein, right? So what we need is we need an increase in calcium levels, and the calcium will then cause the power stroke to occur. So that's when we have shortening of our sarcomere. And once that occurs, the ADP and the phosphate, they're released, and then we go back to this state here called the rigor state, right? And it's called the rigor state because the muscle is still rigid, it's still contracted. We need ATP to relax that muscle, right? So that's why there's like that phenomenon called rigor mortis, where someone dies and their body gets really, really stiff. Because when you die, your body does not make any more ATP, right? So without ATP, you stay in this state here. and you just are really, really stiff. Right? It's really hard to move you. Right? If you've ever dissected a cadaver before, that's the reason why. Yeah. So here kind of talks about that events that we just mentioned. So in order for the muscle to relax, other things that occur like calcium coming off of the troponin. So once that occurs, the binding site on the actin is blocked by the tropomyosin, so that myosin cannot interact with the actin anymore. And what happens to the calcium? The calcium is released and it's pumped back into the sarcoplasmic reticulum. Right, so this is the end of our a very lengthy discussion. But the thing is that there's also a part of chapter 9 that's also muscle anatomy. And so basically for that, you just have to know for the muscle, you have to know the insertion, the origin, and the function of that muscle. And so there's a few things that I encourage you to download to help you with that. The first thing is the list of need to know muscles, right? So on the Moodle, so right after this, I'm going to post this Word document, and it's a list of muscles that you need to know for the origins, insertions, and functions of. And like I'm still revising it, so I'm like kind of removing a lot of muscles so that there's like a core few of muscles that you should know the origins, insertions, and functions of. And there's a PowerPoint that will go along with this list. The PowerPoint's going to have all these muscles in it, so only focus on the muscles that you need to know, right, from the list. So you have a list, and then you have the PowerPoint with the diagrams of the muscle, where it attaches on the bone, and the function and insertions of that muscle. And as you go through the PowerPoint slides, you might notice things like, oh, like nerve innervations and stuff like that. You don't have to know that stuff. Don't focus on that stuff. Focus on the origins, insertions, and the functions of each muscle on this list of need to know muscles. And again, like this stuff here, I'm going to post afterwards, after this discussion, because I'm still like revising that list, kind of making it shorter to make it more reasonable. And on the exam, I can always just give you like a muscle, right? So imagine like my really bad rectus femoris muscle, right? So there's an arrow pointing to it, and I can ask you a question like, okay, what is the insertion of that muscle, or what is the origin of that muscle? Or I can ask you like, what is the function of that muscle? And if you want more practice with this, there's going to be like a practice quiz, so don't worry. There's another thing I want to point out is for the functions, there's like, oh, like flexion, extension, like stuff like that. I encourage you to watch this video so you know what that means. Because if you understand the insertion and the origin, then you're gonna know like the function pretty much, right? Because how it works is that you have an insertion point and an origin point, right? So for example, the origin of the biceps, right, is somewhere in the scapula, right? Somewhere in the scapula. So it's like somewhere up here in the coracoid process of the scapula. And the insertion points of the bicep is in the radius, right? So when the muscle is contracted, it shortens, right? And what happens is it's going to bring the insertion points to the origin point, right? So when you contract your bicep, what is it doing? It's flexing your forearm, right? It's flexing your forearm because the insertion is here in your radius bone. The origin is here. When the muscle contracts, it shortens and it brings the insertion point to the origin point, right? That's muscle contraction for us, right? Okay, so just keep an eye out for this stuff. For now, I think make sure to go over those exercises because again, there's like a lot of physiology. It's very physio heavy this week. So there's exercises and videos help you kind of wrap your head around action potentials and motor neurons. So if you guys have any questions, you guys are totally free to go. If you do have questions, you can stay after and let me know. I can answer those for you. And I think if you emailed me yesterday, I'm just getting to your replies. So I think there's like a few people who are asking me to extend activities for them. So I'm gonna do that after. Yeah. I'm gonna do that after session.
All right.
Cool.
That's my only thing I wanted to add.
Okay, perfect. I'll get to that. Anything else before we depart? Okay.
No,
All
this is
right.
all
And,
high school stuff.
yeah, I mean, some of it is. I think there's a little bit more
Most of
detail.
it, yeah.